To the ABSS webinar series. We are very, very lucky to welcome Associate Professor Xian Wei Che, who actually started his scientific training in Australia, in Monash University, at the Monash Alfred Psychiatry Research Centre, after a Masters of Psychology from Southwest University in China. His work sits at the intersection of cognitive neuroscience and psychiatry. He has publications in journals such as Cell Reports, Medicine, and Clinical Neurophysiology. Today, he's going to tell us about a recent paper published, which is titled Frontoparietal Network Mediates the Antidepressant Effects of Accelerated a-iTBS and a-cTBS, a TMS-EEG study. And yeah, we're really happy to welcome Xian Wei. So before we start, I'm going to acknowledge the traditional owners of the lands on which we meet today. Here in Perth, we are on the lands of the Whadjuk Noongar people. I'd like to also pay my respects to the elders past, present, and emerging of whatever lands that you happen to be on today. So with that, I would like to hand the microphone over to Xian Wei. So yeah, if you want to share your slides.
Yep. Is that okay?
Yep, we can see. Yep.
Yeah, great. Thanks, Lee and Cassandra. I'm really honored to present in the ABS webinar today. I did my PhD in Melbourne in Monash University from 2015 to 2019. I was really honored to learn with my supervisors, Paul, Robin, and Bernie, and also other students and colleagues. And today I'm presenting a study titled the Frontoparietal Network Mediates Antidepressant Effects of Accelerated a-iTBS and a-cTBS. We all know TBS or a-iTBS or c-tBS. So these are some of the milestone works by in a-iTBS. One is from the study by Huang in 2005. This is a study that demonstrate the a-iTBS and c-tBS effects in human brain, I think, for the first time. And there comes the second study on the right panel. It's published by Bloomberg in 2018 in Lancet. In this study, they run a clinical trial. I think it's multi-center clinical trial to compare the antidepressant effects of a-iTBS and with the tradition of the FDA-approved the 10 Hz rTMS. In this protocol, they deliver the a-iTBS in 3 minutes for 600 pulses, compared to the standard 3,000 pulses 3,000 pulses protocol that is approved by the FDA. They showed that these two protocols are equally effective in terms of antidepressant effects. And this is a really milestone work that pushes the a-iTBS to the clinical settings. Here comes another two studies, the SNT protocol developed by the Stanford group. In this protocol, they used a-iTBS to deliver high dose of TMS, high dose of pulses in a very short period of time. In 5 days, they delivered 97 pulses. That is equivalent to the FDA-approved 10 Hz protocol for 6 weeks. So it's a really a high dose accelerated form of TMS, and they generated very promising results, reporting response rate around 60% or something. And the first study, 2020, is an open label trial. It's an open label study. In 2021, they published a small sample randomized control trial. In this year, 2026, they published another study, which is slightly larger than the 2021 study. Starting from 2022, we were working on another protocol, a-cTBS, because in clinical settings here in China, a lot of patients would be treated with inhibitory 1 Hz protocol. So we were thinking that if we could use the a-cTBS protocol to accelerate the depression treatment. So we compare the a-cTBS versus a-iTBS in the group patients. We specifically choose the TRD patients with moderate to severe suicidal ideation, because for this group patients, we generally deliver inhibitory stimulation protocols instead of the excitatory protocols. So we compared them in the small studies with 22 patients in each group, and we can see the results from here. In Figure B, we show the results on depression score. It's pretty much the same as the Stanford protocol, but it's slightly slower. For Figure A and C, we showed the results on the suicidal ideation and anxiety. We can see that a-cTBS is slightly more effective than the a-iTBS in terms of suicidal ideation and anxiety, but it's only at the end of the 5-day treatment, but not in the follow-up stage. In the right side table, we present the response rate data of all these metrics. Especially for the depression, we can see that we have a response rate around 50% mark, which is lower than the Stanford protocol. The generally reported response rate around 60 or even more percent, 6% or even more. It's mainly because that our patients have moderate to severe suicidal ideation, which is a little bit more difficult to manage compared to the patient cohort in the Stanford studies. Aside from the clinical effects, we also record some brain regions, brain activities using EEG and fMRI. Here's an example of concurrent TMS-EEG. The left side panel shows the setup for the technique, and we can see the setup and the big TMS-evoked artifacts compared to the resting state brain activity. In the middle panel, we show some typical artifacts generated by this technique, and when we do all the cleaning, we can see some results. We can see some data from the right side panel. We generally look at the TMS evoked potentials and oscillations, as well as the phase synchronization. Aside from the metrics at the sensor level, we can also calculate the data from the source level, like this one. We can calculate the current scattering and the current density in the help of a personal structure image. So with this technique, we further analyze the data from the a-cTBS and the a-iTBS trial. This is the same data set from the early trial. Here we record the concurrent TMS and EEG, both at the pre and post the 5-day treatment. In a-iTBS, we position the coil at the left DLPFC, because this is the target that we stimulate. In the a-cTBS group, we position at the right DLPFC, and we also have the healthy control data to match the patients in the two groups. This is a slide for the clinical responses. It's actually the same data as the clinical paper, but just with different sample size. In the original clinical studies, we have 22 plus 22 patients, but for this each data, we only have 20 plus 20 for each group, because we have two patients that did not finish the trial, did not finish the clinical trial in each group. So we can see the clinical response is the same as the earlier slide. We then analyze the TEP response at the target region. In a-iTBS, we can see that there is an increase in the amplitude of P100, and in the baseline data in the group is smaller than the healthy control. For the a-iTBS, there's a difference in the N100 time window. Overall, there is a much smaller. There is a smaller absolute amplitude from pre to post treatment. We then move to the source level. Sorry, this is another slide for other components. this is a data for other components that is not significant from pre to post treatment. We then move to the source level. We calculate the current scattering from the target region to other brain regions. We can see that a-iTBS increases some brain responses, especially in the frontal and the parietal regions. For a-cTBS, there's a very strong inhibitory effect in the whole brain regions. We then try to sort out the brain activities using the probability density. In those two groups, we can see that the left IPL region is a region that shows the biggest change. We then extract the data from this region and run some statistics on this region. We can see that it's been significantly changed from pre to post treatment. But with the scattering data, we were not able to build up a relationship with the clinical responses. Here is an example of the dynamic changes of the scattering data. This window shows that the time window that we use to run statistics is a window that shows the biggest change from pre to post treatment, which is confirming the data that the time window that we use to run the statistics in the scattering data. We were not able to build up correlations with clinical responses. We then calculate another matrix called current density, and this time we finally showed that the current density data in the left IPL region is associated with the changes in the depression score. In both groups, the changes in the current density data in the left IPL region is associated with the clinical changes in each group. The current density metric is more like a local brain. It's a local metric, a brain region metrics compared to the scattering data that is more like connectivity metrics. In the right side, there's some regions from other. These data from other regions that is not changed by the treatment. So as a short summary, that we know that we show that in two studies that a-iTBS can be used to treat depression, especially TRD patients with moderate to severe suicidal ideation. And use TMS-EEG, we identified the frontoparietal network that is mediating the antidepressant effects for a-iTBS and a-cTBS. But we know that there are other neurocircuitries or neuroactivities that is being identified for these antidepressant effects, such as the SGCC area and also the insular cortex. In clinical settings, we also feel that the SNG trial is too long for patients. It takes about 10 hours or even more for patients and for caregivers to finish the treatment. So we try to modify the programs with some ideas. In this study, we focused on the dose response curves, because this curve is taken from the SNT paper. It's more like a hypothesis, so it's not generated by real data. We try to work on these curves, and so it's to see if it's informative for us to design new protocols. So we designed two studies. In the first one, we tried to identify the dose responsive curve in healthy controls. So we have two groups, the DLPFs group and the sham group. We ask participants to finish a one-day protocol, which is a 10 a-iTBS session protocol. After each session, we record the brain activity with TMS-EEG. So we have 11 time points in total, along with the baseline data points. So in this way, we can plot the dose responsive curves to see if it's informative for us to design new protocols. In second study, we run a small sample clinical studies to try to validate the results generated from the study one. So we can for the healthy studies, we focus on N100 because this is the most prominent component in the TMS-EEG technique. So we run a whole brain class stats, and we identify the component that is around the frontal and the parietal regions. When we get the data out, we plot them with we try to plot them against the number of sessions. We can see that there's a much quicker change, increase or decrease in the early sessions, and then in later sessions getting more flat, going more and more flat. So we think that there might be this might be the dose response relationship for the single day, at least for the single day a-iTBS protocol. We then move to the whole brain analysis, because in the last slide we focused on N100 component. We then move to the whole brain analysis. We identified another component. It's in the P100 time window. It's still localized in the frontal and parietal regions, and when we plot them out, they look like this one. So it's still in the very early stage. They change rapidly, but for the later sessions, there's a flat line, or there's not much increase or change compared to the early sessions. So from these studies, from those two results, we know that there might be a dose response relationship in the at least for a single day a-iTBS protocol. And these are data from other components that is not being changed, or there's no such a curve. So we then carried out a small sample size randomized control trial. We designed the a-iTBS 6 protocol, which is a 6-session daily protocol for 5 days, compared to the sham protocol and to the SNT protocol for 10 days per day for 5 days. We can see the clinical responses here. For figure A and B, we show the change in depression score. A is the average data, B is the individual data. At least for the a-iTBS 6, is equal effective compared to the SNT protocol. But for the personal data, we can see that there's a more consistent decrease from post to the 1 month follow-up stage. Figure C and D show the response data and the remission data. The response rate was really close to our data and to the Stanford, to the SNT protocol. This time we go to 60%, slightly more than the 55% mark that we observed in the first study. Because in this protocol, in this cohort, we didn't define the suicide ideation. So there's a really big variance in terms of suicide ideation. So we can see a slight increase in terms of response rate. In terms of remission rate, it's not very promising in the post treatment, but in the 1 month follow up, it goes to the 40% mark. So which is still a very good signal. So when we try to summarize all these studies, we can see that the SNT protocol is still a bit long for especially for clinical treatment. If you take patients from the first session to the last session, it would last more than 10 hours per day. So here we try to shorten to 6 to 7 hours per day, and we can see that we build up the dose response curves, and we take it to the clinical state, to clinical translation, and we can see that the clinical effects are still the same, or still being remained compared to the standard Stanford protocol, SNT protocol. So here comes to the end of the slides. It's harder for me to speak for 45 minutes in these few studies. So I want to thank my collaborators and my supervisors. Manly is a very amazing psychiatrist. She's responsible for all the patients that's been tested in this group. She's highly trusted by the patients. And those are my 3 supervisors during my PhD. I did my PhD in 2015, 2019. It's been a long time for me to graduate and do some work here. So here are some doctors that I'm working with, and here's a small team that I have. So I want to thank the a-iTBS webinar for me to present my studies, and I would like to discuss with a lot of guys and our collaborators, people here online that are on some other studies or some on these studies. Thank you.
Thank you so much for an outstanding presentation. We have time for questions. If you'd like to ask questions, please go ahead and unmute. I haven't checked the chat yet. If you have any questions, please go ahead and unmute yourself and go ahead and ask them. Maybe you can stop sharing your screen for now.
Yep.
So I'm just gonna scroll down.
Yep.
Because we have a lot of people in the audience. For example, I think Aaron.
Aaron. I saw Aaron.
Who do the kind of TMS-EEG type of work. It was really maybe I'll go ahead and ask a question. It was really fascinating to see. So you had the comparison between the 6-session and the 10-session protocols, and then in some instances the 6-session one seemed to be more consistent than the 10-session one. Well, I wondered if you could speculate for me why that might be the case. Is there some type of point of diminishing returns?
Yeah, when we saw we tried to consider this from the dose response curves. If you see here in the very late sessions, it might be not be additive effect on the cortical responses, maybe even in the reverse direction or in the wrong direction. So they might be associated with the responses we observed here in the clinical data. That is, the 6 sessions is more consistent in terms of the change score in depression.
This might be a very pedestrian, very, very simple question, but because this was said 10 sessions within one single day, could there be time of day effects as well that limits the efficacy of a 10? Is this because this is 10 sessions within one day, right? So would it be later? But yeah, it's really fascinating to see this type of dose response effect within a single day. And I think, it seems really promising given how consistent, how tight that data looks.
But it's unfortunately that the data has been keep being rejected by many journals. It's a little bit unfortunate, but it's okay. I think one day would be accepted in some decent journals.
Yeah. Is questions from the audience? Yep, go ahead.
Hi, Shenwei. Great to see you.
Hey, Aaron.
Fantastic talk.
Thanks, Aaron.
I'm up on the big screen. I don't really have many questions. That was really interesting. I was just curious about the sort of the protocol you were running in terms of, and sorry if I missed this because I didn't catch all of your talk, but in terms of tolerability and did patients generally find it fairly tolerable? Did you have many adverse events or any adverse events?
Yeah, great. Yeah, thanks, Aaron. We collect the data of the side effects, but I have got to put it on the on the slide. Sorry. Yeah, it's okay. Compared to the S&D protocol, I think it's fine. We found in clinical settings that some people that like the 10-session protocol, they think they're being treated much longer time or much more pulses. But for a lot of patients, they like the little bit shorter version, because in clinical settings here, SNT is generally being used for studies, not for clinical treatment, because it's really long, and it's really time consuming, and it's really expensive actually. Here you have to pay to get TMS treatment, and we try to cut it short. And when we see the side effects data, it's okay. It's similar to the SNT protocol, because we have run 3 to 4, I think 3 to 4 SNT protocols now in different patients. They are okay generally. So I guess it's okay for them, and so we try to make it more comfortable, or at least more affordable for patients, or less expensive.
Yeah. Oh, that's good to know.
Yeah.
Questions from the audience? What I might quickly do. Yeah, sorry, I was just clicking through speakers to check if there were questions from the audience. So if I understand it correctly, the for your, say, your 10 session 10 and 6 session single day design, it would have been the EEG setup would have been done just pre and post either 10 or 6. Is that correct?
Yeah, for the healthy studies, we collect data, we collect each data for many times in healthy controls. But for the patient study in study 2, I think we only collect data for in only a few patients, or around half of the patients at baseline and after 5-day treatment. So we were not able to present a really strong EEG data in the study from study 2. We try to do that, but it's really hard for us to get for all the patients to be consenting or to be able to willing to go to EEG testing. So it's not a it's not like it's only for the study, so they are willing to or not willing to be tested. We have like so we have 15 patients in each group. We have like 6 to 10 or something each data for patients. So we're still working on each data, but it's still hard for us to get something really solid from such a small sample.
I think it was still really impressive that you managed to get, this is not an easy protocol. It's really outstanding that you managed to get such a big involvement of patients within this both EEG and TBS protocol.
Yeah. One critic that we received from the study is that we were not able to build up the curves from the patients. So it's really hard for us to have patients to test so many times, even for them to have a baseline testing and for post-treatment testing over 5 days. It's difficult, especially for TRD patients. So we did the healthy studies first, and still it's I think it's a limitation that we were not able to present data all from the patients. So we choose this one to go through healthy controls.
We have a question from the audience from the chat. Alex Tang asks, for your dose response, you found changes to some TEP components, but not others. In terms of plasticity, what do you think the rTMS is doing? And is it changing specific circuits and cell types? I don't know if you're familiar with Alex's recent work, but it's kind of relating to the work that he's done recently in animals that looks at circuits and cell types.
Yep. Thanks for the question, Alex. It's really hard for me to know the plasticity data because I'm not an expert on animal studies. We're running some studies on animal TMS, but it's still in the early stage. We tried the a-iTBS and a-cTBS and 10 Hz protocol in rat with TMS, and we saw some effects from I think the most promising one is still the a-iTBS. Then comes a-cTBS to 10 Hz. I think 10 Hz is the least effective one compared to the other two a-iTBS or a-cTBS protocols. But still, in different data sets, we generally see different responses in different components, such as early components or late components, and also hard for us to really tell what is really generating or what neurochemistry or neurotransmitters are really generating the components of the oscillations. What we do now is that we have a sham coil now. In the early days, we didn't have, so we tried to put a sham coil on the studies to say that if the results or the components, either the components or the oscillations, are really unique to the real coil. So in the data set, we actually had the sham data too, but only in a few patients, I think in a few participants in the first study. We can see that some of the components are really unique to the real coil, not the sham coil. So my answer is that it's really difficult for me. It's really hard for me to understand really can tell that the neurotransmitters or neurochemistry responses behind the components or oscillations. So we're trying to work on that, and That's my answer.
Maybe you can speculate on for us, because it sounds like
Yeah,
because
yeah,
Wei is also doing animal work. Alex, yeah,
yeah,
if you, I know you're in the lab, but Alex is typing probably because he's in the lab and can't talk. When you're able to, if you wanted to
yeah.
type your speculations in the chat based on what you know in animal work, where perhaps Wei and their lab could look at in their animal work. Yeah, it would be fascinating to hear what you think, Alex.
Yeah, so
Um,
I have something to add on from these results from the data too. So here when I analyzed data, I thought that would be so people say that a-iTBS is excitatory and a-cTBS inhibitory. So when I see the data from here, it's a bit promising that I see that the a-iTBS increased the brain responses, but a-cTBS is decreasing. But there's argument that if the amplitude of 100 is like a is bigger or better or smaller better. There's still argument on that. And we then move to the to the source level data. We can see that we can pretty much guarantee that a-cTBS is very inhibitory and a-iTBS is very is excitatory, at least for or some of most of the brain regions, or at least for the regions that is connected to the target region or the frontal cortex. So that's my experiences or my speculations from the two data sets that I can generate.
can you speculate for me about, or maybe inform me, because I am relatively, I don't have a lot of knowledge about this. But the I guess the mechanistic rationale for having different hemispheres
Yep.
for cTBS and a-iTBS and a-cTBS. Why different hemispheres?
The reason that we position that there's an argument that the depression paper have a like patients have a imbalance of the two hemispheres, like left side and right side. Right side is overall like more or highly or more excited, and the left side less less excited compared to the healthy controls. So for the patients, we used to we usually we generally deliver like excitatory protocol for the left side of the left brain region to increase its excitation. And for the right side, we go to inhibitory 1 Hz stimulation, or we go to cTBS or a-cTBS to decrease its overall excitation or increases inhibition. So to make like a more balanced hemispherical interaction between the two sides. That's a rationale for us to for in clinical settings for us to treat patients, or for this study that we decide to go to left side excited and go to right side inhibitory.
And the dependent variable. Yep. Sorry.
Yeah.
And one of the I guess it was interesting. Yeah, one of the dependent variables for the cTBS right hemisphere manipulation is the measurement at the left hemisphere. So yeah, thank you.
Yeah.
So Alex has a follow-up. So he says, I'm just curious that when you see changes to some components of TEPs, but not others, what that means
Yes.
for circuit function. Based on the TEP changes you see, does that match with TEP changes seen in depression, i.e., is accelerated rTMS targeting the same circuit functions that are abnormal with depression?
Yep. Yeah. Great question. For us to is that we can see that the patients here, we show that the patients have when we compare to healthy controls, they have like impaired responses in the side, in the left side or in the right side, but in different fashion. We generally don't to my understanding, or in our lab, we generally go with sensor level statistics, like especially for TEP, we consider it as a local or brain responses. We don't go with speculations on like circuitry level. So we limited the TEP response to like a really local responses to the treatments. When we want to go to circuitry or go to go to other brain regions, we generally choose oscillations, or we choose to go to source level analysis. We can do source level uh, analysis and to decodings to see which brain region is generating the oscillations. but it's still not very accurate compared to functional MRI. In terms of neurocircuitry evidence, we prefer go to fMRI now. We also have an fMRI data for the study. I did not present here, but for the fMRI data, we can see that there's circuitry changes, including we compare the patients to health populations first, so we can see the abnormalities or abnormal neurocircuitry evidences in some circuitries, including the frontoparietal network, and also including the frontal insular network, which is also a very promising neurocircuitry or target in depression treatment. And then we can see that the a-iTBS and a-cTBS can both change these circuitries. So it's really hard for us to go to have really strong neurocircuitry evidence based on EEG or based on TEP. So we tend to go to source level evidence, or we tend to go to functional MRI to go for more like a solid or go for stronger neurocircuitry evidences. That is what we do in our lab. Thanks.
Thank you. Yep. Yeah, so Alex has follow up. In case it's useful, and I'm reading these out because in the recording chats aren't recorded. So Alex follows up that in case it's useful, it was recently shown that accelerated a-iTBS acts via intrathalamic neurons, not pyramidal tract neurons. So from your EEG data, it could be interesting to look at brain regions that are connected via intrathalamic connections.
Yeah. That's a great idea. But we generally get criticized by EEG data that when we go to this deep, or we go to thalamus, we go to even deeper regions like the PEG regions. A lot of critics that are being raised by sometimes to our papers or to our studies, that we wouldn't be able to analyze this deep, or at least in a very accurate way for these deep regions. So it's hard for us to really put up a strong data evidence based on each data. We then try to merge. We then try to go with a functional MRI to combine them with EEG. I think that's a very promising technique, especially with high density EEG. We're now working on a 128 channel EEG with personal structural MRI data. We try to compare we try to combine the neurocircuitry evidence with the oscillation evidence in this setup. So that would be a good idea, but it's still a lot of very challenging for each technique to go to this deep or go to these small regions. Thanks, Alex.
Yeah, that's yeah. So just reading it out, lots of critics about cellular and circuit work too. Yeah, absolutely. Do we have any further questions from the audience? I might ask a quick question if that's okay.
Yep.
I guess, theta burst stim with TMS is an obviously much more well-established way of modulating brain excitability. I was wondering if there is any interest to use other forms of brain stimulation than the kind of newer forms of brain stimulation, and what kind of limitations those might have. So the examples I'm thinking of are like transcutaneous vagal nerve stimulation or functional ultrasound stimulation. Is there in where you are is there interest or are there barriers to trying these other forms of brain stimulation techniques to treat treatment-resistant depression?
Yeah, thanks. That's a really fantastic question. People are working on different techniques now. TMS is a really superficial stimulation technique. It only goes to very shallow brain regions, but it has a really consistent, really good effect on a lot of at least for a few conditions. People now are working on deep brain regions techniques like ultrasonic stimulation that can go to really deep, and also working on like TIS, temporal interference stimulations, which is a new technique that have been developed by 2017, I think. We're working on the two techniques too, but it's still very hard for us to find some effects on patients. We did studies in health controls, and we can see we work on pain studies because I've been working on pain for a long time. We can see the analgesic effects of those two techniques, especially when we go to deeper regions. But we're not trying to find the conditions that can be for patient population can be useful. We test for TIS. We tested for a few patients with a symptom called MIRG. MIRG is like MEG, so it's a really hard condition to manage. It's very similar to Parkinson's disease, and we stimulate the SDN, which is a really small and really deep regions, but we didn't observe any positive effects. Also, we are working on some neuropathic patients with TIS. Still, we couldn't find some effective, at least parameters protocols of a specific type of chronic pain populations. So those two are very promising deep brain stimulation techniques that we're also doing some work on. But we are still in the health control stage, or still in the experimental stage, that we're not able to, at least in this stage, we still haven't found the very good or very effective condition to be for this to be applied to. For depression, we have a trial ongoing with TIS, the temporal interference stimulation, and we target different regions. But I'm only a collaborator or a also of the project, that we saw some effects with deep brain regions with TIS in TRD patients. I think the trial is still ongoing. We targeted the amygdala, and also the I think amygdala and the posterior thalamus or something, and we saw some positive effects in patients. So we're still working on the trial, try to make it more effective, or at least for these two techniques to find something that to be effective for those two techniques. That's what we're working on.
Yeah, it sounds like, I mean, it's really outstanding that you're, very rapidly taking this to the clinic, not just looking at healthy control populations. It seems like also none of the clinical populations that you are tackling are easy populations. These are, even your depression studies are all conducted in really treatment-resistant, suicide-prone individuals. So yeah, it's been really fascinating to hear from you about the work that you are doing. Do we have any further questions from the audience? In Australia, at least I only know of, maybe people in the audience can correct me if I'm wrong, but yeah, I don't think there are a large number of groups doing those techniques in these types of clinical populations at scale yet. So yeah, what you're doing seems very much at the cutting edge. So if people have no further questions, I might go ahead and finish that up for today. If anyone in the audience, again, as always, if anyone in the audience has new work that they'd like to present, please let me or Cassandra know. As we've mentioned before, we will upload these on the a-iTBS, the Australasian Brain Stimulation Society YouTube channel, which means that it can reach a larger audience beyond the people who can make this specific time of the year, specific time of the day. It is a slightly busy time for us who are doing teaching here in Australia, hence the relatively smaller audience. For example, I'm finalizing marks for my teaching unit now. But yeah, thank you so much for this outstanding presentation. I really enjoyed it, and hope everyone else here has had fun. We're going to make sure that the ABSS community also sees that this video has been uploaded, such that everyone can engage with this outstanding work. Thank you so much again, Xian Wei. And without further ado, we'll finish up. Yeah.
Thanks.
Thank you so much.
Bye bye, guys.
Thank you. Thanks. Bye.
Be good. Bye bye.
Thank you.
Thank you. Thanks. Thank you.